Buenas noches. Good evening, everybody. Welcome to day 2 on week 16 of the semester. I'm very happy to see you guys here, as this is going to be our last official day of lecture. Thank you for always coming and participating in class. Hi to Maria on the chat. Hi, Morea. Hi, Kiyomi. Really great to see you guys. Hi, Elizabeth. Hi, Guadalupe. Thank you for coming, you guys. Now, hi, Katie. So we are almost done with the semester. Today is going to be the last official lecture. So we're going to wrap up the concepts of chapter 10, and that is the last concept. Hi, Dominique and Nikki. These are the last concepts that are coming in the final exam. So I actually put together the study guide. I just need to finish up highlighting the items that are coming. Understand that the final exam is cumulative. It's going to open for 24 hours next week on Thursday. Our last meetup together is actually going to be lecture next week on Tuesday. I will be holding office hours next week on Tuesday. That being said, I also posted chapter 10 homework and it's due on Friday. The last lab assignment that you guys have, which is the synthesis, not the synthesis, the isolation of casein. Remember that it's actually due this Saturday. If by, any chance anybody gets sick, they need an extension on the homework, no problem. Email me and I'll be more than happy to help you guys. Now, understand that I am limited by the deadline when classes are over. So make sure that you hand in all of the work that you have due by next week on Friday, because pretty much after that, the semester is over and I'm going to have access to Canvas, but it's going to be limited. And then after that, I have to hand in grades. So understand that so far you have your last lab that you need to hand in by Saturday. You have chapter 10 homework. You have the My Favorite Molecule project, which I already put in what I'm expecting of you guys. Basically, I gave you some ideas of molecules that you can look into or sites where you can look at organic molecules. You pick a molecule and then you answer the questions. Then you submit to me a single PDF file, and that's going to be the project. So that's pretty much what we have going on from here to the end of the semester, okay? Any questions before we get started? Oh, if you go to Canvas, the assignment for My Favorite Molecule project is already being assigned. Let me open my Canvas site for the course. So you guys see where it is available. Give me a moment before I share my screen. Student view. Okay, so understand that you guys have, these are the assignments that pretty much we have other than the final exam. Chapter 9 quiz, which is due tonight. Chapter 10 quiz, which is due next week on Friday. And here you will have My Favorite Molecule project. When you click on it, you can see that it says PDF file with the questions, and you can see that it says instructions. Then it says PDF file with ideas. And as you can see from the instructions, you have to submit a single document, PDF format, with the answers to the following questions. And as you can see here, this is basically the rubric of how points are going to be assigned to each of the questions that you have here. The My Favorite Molecule project is actually 25 points and is actually 5% of your grade. So as you can see, you have until December 16th to update that file. So for example, on your end, you just click Start Assignment. And when you click Start Assignment, then you can upload the file and then you submit the assignment. So that's pretty much what we have. Okay. Is anybody having the similar issue that the assignments are not showing up? Can somebody verify if the assignments are showing up? Because in my student view, they are available. Can anybody corroborate that the assignments are available for everyone? So for the people that are able to see it, you guys are able to see the labs, the 2 chapter quizzes, and then My Favorite Molecule project? You guys are able to see all of the assignments that are due from here to the end? Okay. So maybe at the end of class, I can help the student that is not able to see it. It depends if you are trying to reach it on a computer versus a tablet, because sometimes, oh, okay, it's working out. Great, great. That's great to hear. Awesome. No problem. I prefer that, everything is working out and then you guys are able to see all the assignments that are due, because I don't want anybody to be missing the work that needs to be submitted. Now, I'm just going to mention it again. Understand, you guys, that when it comes to the final grade, Canvas is what I utilize to store all of the grades. When it comes to the final grade overall, remember that in this course, you guys get to drop an exam, you get to drop 1 overall lab. In addition, you guys have extra credit points. Canvas at this moment is just illustrating all of the grades without taking into consideration the things that are going to be adjusted for your final grade. So even though Canvas is able to give you the grades that they're showing you there, it gives you an idea overall of what your grade is going to be, but it doesn't give you the whole view. I actually do not change all the grades that are on Canvas. I actually extract all of the numbers and utilize the numbers to recalculate your grade. Understand that your grade is going to be submitted through the system that Maricosa College has for grade submission. But in terms of the numbers themselves, understand that I have to make adjustments based on what was said in the syllabus. So please don't be concerned when it comes to like, oh, is my extra credit really extra credit? It is. It's just that Canvas is not able to make those adjustments. I have to actually look at case by case and make those adjustments. So please be patient when it comes to the final grade. Grades, I actually cannot email students what their grades are, because we actually at colleges have rules that prevent us from doing that. But be sure to understand that I always have my students' best interest at heart. And I actually sit down and look at each student's case, make sure that the calculations are up to par. It's just that Canvas as a whole is a way that we can store grades, but the calculations, I personally do them, okay? So next week, we're going to review the concepts that are coming in the exam. And then on Thursday, you guys will have the whole day. And at some point during that Thursday, please go over your exam and submit it, and make sure that all your assignments that are due next week, you will submit them. So that's pretty much what we have for the rest of the semester. Now, taking that into account, let's actually, let me get started, share my screen. And taking this into account, let's talk about the last topic that we mentioned on Tuesday, which was the whole idea of, since we know that amino acids have to come together in the condensation reaction that is called amidation, once they do that, then what we form overall is a protein. Now, when proteins have less than 50 amino acids, that is called specifically a polypeptide, because essentially we have multiple amide bonds. And since amide bonds is what represents the peptide bond, that's why anything shorter than 50, and especially if they don't have any biological function, we call peptides. Now, in class on Tuesday, I illustrated to you guys how, and you will see that in chapter 10, chapter quiz, you will have a series of exercises in which you will have a peptide, and then you have to determine the one-letter codes for that peptide. As I mentioned to you guys, please make sure that when you do this problem, you do it carefully, because as an incentive for you guys to do these problems, understand that the peptides are going to specifically spell words. Now, some words are going to be in English, some words are going to be in Spanish. So understand that this is just an incentive. So at least you know that if you're not spelling a word, you'll be like, okay, wait, this word, so this peptide, I may not be assigning my amino acids correctly. So taking this into account, I want to do at least one more problem, so we, again, review how to determine the sequence of amino acids that are found in a peptide. So here we have the following peptide. As I mentioned to you guys, when we have a peptide, they always initiate with an NH3 group, okay? That nitrogen is going to be positively charged. Unless your first amino acid is proline. And when we look at the amino acids, here is the structure of proline. For proline, it's the only amino acid that is not going to start as NH2. It will that it will not start at NH3. Is going to start as NH2, because proline is the only amino acid that the side chain, which is this area, is actually connected to the amino. So remember that that's just special for proline. Now, taking that into account, when we have a peptide, give me a moment. There is always a trend or a specific repeat that we see in these type of structures. And the repeat is as follows. You start with an NH3. Immediately after the NH3, you're going to mark the carbon with the hydrogen, because that's going to be the alpha carbon. That alpha carbon is going to contain the side chain that represents the amino acid that we're looking at. Then after that, we can observe that we're going to have amide bonds. Followed by the amide bonds, we're going to have another alpha carbon. Amide bond. Alpha carbon. Amide bond. Alpha carbon. And this pattern is going to continue between amide bonds and alpha carbons. I'm going to keep on highlighting them. Now, after an alpha carbon, if you see a C double bond O, O negative, that means that that's where the peptide concludes. So in the following peptide, all the highlighted carbons that are in blue, that represents the alpha carbon. So we're going to look at the side chains that are pointing down or up from the carbons that I highlighted in blue, because that was what represents the different amino acids that we have present. So how do we find out what are the amino acids that are present? Well, we look at the side chain. So for example, here, I'm going to use my highlighter tool. Here we have a CH2, 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 NH. It has this group. This is one of the basic amino acids. So when I look here, oh, let me move the image so we see all the amino acids. We can actually see that that side chain is arginine. Okay. Now, arginine, even though in this figure, specifically, you don't see the 3-letter code and the 1-letter code. Let me show you. Here, we can clearly see that arginine has the 1-letter code R and the 3-letter code ARG. But right now, we're just using the 1-letter code. So that means that my first amino acid in my peptide is going to be represented by the letter R. Let me use the highlighter tool. Then I go to my second amino acid. This is a CH2, CH2, and I have a carboxylate. So then I go here. CH2, CH2, and then I have a carboxylate or carboxylic acid. Well, that is glutamic acid. Okay. Glutamic acid has the 1-letter code E. Then I have a CH2 with a carboxylate. Oh, the CH2 with a carboxylate or carboxylic acid, that is aspartic acid. This has been misspelled. This is aspartic acid. And when it comes to aspartic acid, the 1-letter code for aspartic acid is D. Next, I have a CH2SH. CH2SH is right here. That is cysteine. Cysteine has the 1-letter code C. Then I have a bond here that it looks in dashes, but that's just a methyl group. Let me use a different color because that's really close to the one that I use for cysteine. So this one is just a methyl group. And the amino acid that is just a methyl group, you can see here that is alanine. The 1-letter code for alanine is A. And then lastly, you can see that we have another arginine. So that means that we have R again. So this specific peptide, as you can see, is REDCAR. So this is just, is spelling the word red car as one word, okay? Just make sure that whenever you are putting your answers, remember, don't put spaces between each of the letters in your peptide. Any questions when it comes to given a specific peptide determining the sequence of the amino acids? If you're ready for the next topic, put a 1 in the chat. Really quick, there's a question on the chat. The letters actually come from the symbol that is utilized to represent the amino acid. As you can see, glycine is represented by the letter G. You can also represent glycine by the letters GLY. Alanine is A, valine is V, leucine is L, phenylalanine is F, tryptophan is W, so that's where it comes from. The image that I have over there that I was utilizing as a guidance doesn't give you the 1-letter codes and the 3-letter codes, but this figure is also on the slides. In the internet, you can actually find images of having skeletal structures of the amino acids with the full name, the 1-letter code, the 3-letter code. So I provide to you guys different images when it comes to the amino acids. Use the one that makes sense to you. Excellent. Let's now move on to talk about the next concept when it comes to proteins. Because whenever we were talking about the sequence of amino acids in a protein, that is known as the primary structure for a protein, okay? Now, proteins have, in addition to primary, other organization levels. Those organization levels are secondary, tertiary, and quaternary structures. The first one, or in a way, the second one that we're going to discuss is the secondary structure for a protein, okay? Now, as you can see here in the slide, the secondary structure is the 3-dimensional form of local segments of proteins. What does that mean? That we already learned that in the primary sequence of a protein, we are going to have amide bonds that are present. And these amide bonds that are holding those amino acids together, let me just illustrate it by showing you guys the structure. So the amide bonds that you see here highlighted in yellow, they can actually interact with each other, okay? Understand that in biochemistry, we have what is called secondary structure propensities, which means that there are sequences of amino acids that tend to form a particular shape for secondary structure. And that's how we know which one of the secondary structures will be formed within a protein. Now, it is important to note that at least for the discussions in this class, that when it comes to secondary structure, you can form one of 2 3-dimensional forms. You can either form an alpha helix, which as you can see here, it looks like a winding staircase. And then we could also form or proteins can form a beta sheet. A beta sheet is just like a pleated way that the peptide can actually shape itself, or the protein for that matter. Now, how do these structures actually form, the alpha helix and the beta pleated sheet? As you can see here, they are formed by hydrogen bonds between specifically the carbonyl oxygen atom and the hydrogen in the amide groups. So what that means is that if I have a particular peptide, specifically the oxygen is going to be interacting specifically with the H in an amide, okay? Not the side chains, is going to be in what we call in biochemistry the backbone. The way that I illustrated here in between one carbonyl and a nitrogen down the road is because typically we see these interactions happening between 3 to 4 amino acids in length. Okay, so these terms are going to be separated by 3 to 4 amino acids in general. But understand that the secondary structure gives these two forms of folding in a protein. Now, in addition to this, because the folding, remember that for the secondary structure is just the amide bonds in the back, the side chains of the amino acids can also actually give rise to different types of interactions. So in the tertiary structure of a protein, understand that this specifically is an overall three-dimensional shape, but it is important to know that it's caused by interactions of different parts of the chain. What parts of the chain are involved in the tertiary structure? The R groups, meaning the side chains of the amino acids are now going to be interacting with water or with each other to give rise to the tertiary structure of a protein. As you can see in the final in this figure, we have a list of tertiary structures that are going to be occurring in a protein. And I'm going to go through them one by one, starting with a hydrophilic interaction with water, okay? So some side chains of amino acids actually can have a hydrophilic interaction with water. So the parameter that these amino acids need to meet is that the side chain must be a polar side chain. So you can see here, we have examples of tyrosine, that's why I put the letter Y, and serine, that's why I put the letter S. So those are the side chains for those amino acids. They're part of the amino acids that are polar, and that's why they have hydrophilic interactions with water. Now, amino acids can also have hydrophobic interactions. And these hydrophobic interactions are going to happen between side chains that are non-polar. So we can see here two methyl groups interacting with one another. These side chains are actually part of alanine. That's why you see me writing the letter A. The CH2 benzene is also a nonpolar side chain, and this specifically is present in phenylalanine. That's why you see me label with the letter F. I'm putting just the one letter code for the amino acids. So if you see two phenylalanines interacting with one another, that is a hydrophobic interaction. Then if you see two alanines interacting with one another, that is also a hydrophobic interaction. But for a matter of fact, as long as you have two different nonpolar side chains interacting with one another, those are hydrophobic interactions. It is important to know that when it comes to hydrophobic interactions, because those side chains are nonpolar and they are afraid of water, we typically find them inside a protein, meaning they're going to be hidden away from the water that may be outside of the protein, okay? So usually these hydrophobic interactions are inside, they're just inside of the protein structure, folded in there because they don't want to be seeing or interacting with water whatsoever. The other type of tertiary interaction that we can have is disulfide bonds. Now, disulfide bonds are going to exist specifically if we have a cysteine residue. So we have a cysteine residue reacting with another cysteine residue. The side chain of cysteine is specifically an thiol group. And those can actually oxidize to give rise to this disulfide bond. So the C letter that you see here, that's the one letter code for cysteine, okay? So that's what I'm illustrating here. So two cysteine residues are going to be forming a disulfide bond. Now, if we look overall in the following figure, we have several examples of hydrogen bonding events happening. You can see here that we have an example of hydrogen bond, an example of hydrogen bond, an example of the hydrogen bond. Remember that hydrogen bonding events are going to be occurring between an electronegative element that must be nitrogen, oxygen, or fluorine. Remember that hydrogen bonds need to happen with fun elements. Now, they're going to be interacting with a hydrogen molecule as long as that hydrogen molecule is also attached to one of those quote unquote fun elements. Again, fluorine, oxygen, or nitrogen. Now, in amino acids, we don't have the element fluorine. So for the most part, you will see that the attachment of that hydrogen is going to be to either oxygen, as you can see here. The hydrogen is attached to oxygen and is interacting with oxygen, or here, we have an oxygen atom that is interacting with a hydrogen that is bonded to nitrogen. So that's what's going to specifically illustrate a hydrogen bond, okay? Lastly, we have what are called salt bridges, which are right here. And in a salt bridge, understand that this is a quote unquote ionic interaction, but you would know that a salt bridge is actually occurring because here we're going to have a basic amino acid, just like NH3, which is representing lysine. The one letter code for lysine is K, and that can interact with a acidic amino acid. So for example, aspartic acid, which has the letter D as a designation for the amino acid. It could also happen with glutamic acid, which is the letter E designation, okay? But as you have to have a basic amino acid with an acidic amino acid, okay? And these are the different types of interactions that are going to happen with the side chains of the amino acids in a protein, and is what gives rise to the tertiary structure. Any questions on primary, secondary, or tertiary structure for a protein? Excellent. Since there are no questions, let's understand that the majority of the proteins out there have a primary structure, a secondary structure, and a tertiary structure. There is a limited number of proteins that actually have what is called a quaternary structure. And the quaternary structure is going to be present only in proteins that they need 2 or more units to come together to give the full structure of a protein. To give you guys an example of a protein that actually does this, it's going to be here, hemoglobin. So what you guys are actually observing right now is called a ribbon diagram. Ribbon diagrams are actually utilized to represent the structure of different proteins. Understand that if you look under the microscope at a cell, you don't see these ribbons. This is just computational work, and based on different calculations and predictions, and also experimental data, we actually can determine how would a protein actually look like. It is known that for hemoglobin, there's going to be 4 units that have to come together in order to give the overall structure for hemoglobin. And you can see here that 2 parts are labeled in orange, which that is just a false color. That's just the taste that the person that built the structure wanted to give it. And then the other 2 that are kind of like that maroon color, those are the other units. So for example, hemoglobin has 4 things that have to come together in order to give rise to the overall structure of the protein. As I said, not every protein has multiple subunits that makes them come together in order to give the overall structure, but some do. And those proteins are known to have what is called a quaternary structure. Now, one of the things that proteins can actually have is that depending on different treatments, even though they're, as you can see here, nicely folded, unfolding can actually also occur. In section 10.4 in the textbook, there's a discussion about protein denaturation. And by definition, protein denaturation is just going to be a process that results in protein unfolding. Now, understand that the unfolding of the protein is just the result of the different agents that can be utilized in order to make this happen. And specifically for us, we have actually utilized protein denaturation. For example, as you can see here in the following table, to actually resolve some issues that we have had, or actually to create certain products that we consume. So for the final exam, I need you guys to be familiar with the following denaturing agents that exist out there. So as you can see here, the denaturing agents that you need to know include heating a solution that has proteins above 50 degrees Celsius, adding acids or bases to a protein solution, adding organic compounds to a protein solution, adding heavy metal ions to a protein solution, and lastly, oh, I mislabeled this. This is number 4. And lastly, number 5, agitation. Okay, so let's go through each of these. The first one, heating something above 50 degrees Celsius. Understand that in this process, the bonds that are going to be disrupted in the protein are going to be the hydrogen bonds and the hydrophobic interactions that happen between nonpolar R groups. So when you heat a protein solution, understand, well, if you heat it, or if you have an organism that has proteins and all of a sudden is in an environment where we heat it a lot. So for example, whenever we're cooking food or whenever we are autoclaving surgical items, heating something above 50 degrees Celsius, because those bonds are going to be disrupted, what results is that we are going to be affecting the secondary structure of the proteins and the tertiary structure in the protein. Okay, so specifically for autoclaving, that is one of the ways in which you can actually sterilize surgical items. And understand that that is crucial, because if somebody is undergoing surgery, you want those instruments to be sterile. So utilizing this technique, because we're denaturing the proteins, that may result in an organism, for example, bacteria, if it's around, to be now non-functional in its proteins, and it may result in its death. So that's why we do that. For example, if we actually use acids and bases as a denaturing agent, you can see that the bonds that are disrupted are going to be the hydrogen bonds between polar R groups and the salt bridges. So that means that this type of denaturing agent affects the tertiary structure of a protein. Where do we see an example of utilizing acids and bases for denaturing proteins? Well, in the formation of products like yogurt and cheese. So overall, when those products are created, understand that bacteria that is utilized to make the yogurt and the cheese produces an acid, and that's what actually denatures, in the case of milk protein, those proteins that exist in milk. Next, we have organic compounds. When organic compounds are actually exposed to proteins, you can see the hydrophobic interactions are affected. So that means that we are affecting the tertiary structure of those proteins. The use of ethanol and isopropyl alcohol, disinfecting wounds, or during recent times during the pandemic, we might have been using hand sanitizer often. Understand that the idea was that these organic compounds may be disrupting the proteins that may exist in some organisms. Next, we have the heavy metal ions. Heavy metal ions, including silver, lead-2, and mercury, affect the disulfide bonds in proteins by forming ionic bonds. So that means that specifically the tertiary structure of those proteins are the ones that are affected. Now, where do we see examples of that? Unfortunately, sometimes people get these heavy metal ions. One example is in water. So if water is not properly purified, these ions that create poisoning are present there. And if people ingest that water that is contaminated, now you understand that, that can lead to the creation of certain diseases, because now we are affecting the tertiary structure of proteins. Lastly, we have agitation. And when it comes to agitation, it actually disrupts hydrogen bonds and hydrophobic interactions, specifically by stretching the polypeptide chains and disrupting stabilizing interactions. So what this means is that we are affecting the secondary and tertiary structure of proteins. Understand that if any protein of interest has a quaternary structure, as long as tertiary is affected or secondary also, it will also affect the quaternary. I'm just putting it there just because quaternary structures are not as common. But lastly, agitation, an example of that process that leads to protein unfolding include when you agitate egg whites to make a meringue. So, and for those of you that have watched cooking shows or are baking enthusiasts, you know that egg whites, if you agitate them, okay, with a whisk, it can actually create that cloud. And overall, what you're doing is unfolding the proteins that are in those egg whites. Understand that, these are the different denaturing agents that can happen to protein structure, okay? Now, in the textbook on section 10.5, there's a description about protein functions. So proteins can be classified depending on their function. And you can see in the following table, what are the different classes of protein and how we can actually have descriptions for those protein functions. So I want to, as I've done throughout the exams, please just have this table available. You're going to utilize it, because if I give you the function of a protein based on the description that we see in the second column, we can actually classify that protein. As a matter of fact, you guys should also have this table available. So make sure that you have these resources when you're taking that final exam. Now, let's move on to section 10.6, where we are going to be focusing in one specific class of proteins. As you can see here in the function table, enzymes are going to be catalyzing biochemical reactions. So even though there's different classes, we're going to dedicate some time to specifically enzymes, because when it comes to chemistry inside cells, enzymes are going to be the main proteins that are going to be acting on different reactants to make products and other reactions inside cells. As you can see here, enzymes can be defined as proteins that act as biological catalysts. When it comes to the shape of an enzyme, understand that the enzymes, because they're proteins, they're going to have their body, okay? And then there's going to be an opening specifically in that enzyme that is called the active site. The active site of a protein specifically is where chemistry happens. So if we focus on that opening in our enzyme, we can see that the molecules that those enzymes are going to be interacting with are going to happen at the active site. Now, so you guys are not get confused. In biochemistry, we don't call the molecules reactants, we call them substrates. So when you hear the term substrate, understand that this is just another word for reactant. It's just that in biochemistry, we call them substrates. You can observe here that in the active site, which is what you're seeing, so the white area, you know, that is in the middle of that gray area. So this is the enzyme. And then that white area is going to be the active site. The reason why that substrate is able to sit there is because, as you can see, there's going to be some interactions between the side chains of the amino acids and that substrate in order to keep the substrate there and eventually, as you can see here, converting the substrate into product or products, okay? Now, why is it that we that in the body we have enzymes? Remember that since enzymes are going to be proteins that are biocatalysts, their role is to speed the rate of the reaction. Just like the catalyst that we have observed before, which we mentioned many weeks ago of what is a catalyst. Overall, what's going to happen is that enzymes are going to make sure that the rate of the reaction is increased by doing one specific action, and that is lowering the activation energy. So in this figure, we can see the following reaction. When CO2 and water come together, we actually make bicarbonate and H+. So the line that it is in that minty green is just illustrating what will be the overall changes in energy as those reactants turn into product. Now notice that specifically, let me just do some lines here. Give me a moment. So without an enzyme, we can see the trace of how CO2 and water turn into bicarbonate and H+. But now, when we have an enzyme in the system, we can see that now our activation energy is lower. So the hill is lower, and we're able to reach faster to the products of the reaction. So remember, whenever we have a catalyst, we lower the activation energy, and that results in the increase specifically of the rate of the reaction. You can see it here in words in the second bullet point. Now, these enzymes have a variety of names. So the names of the enzymes come from, for example, the type of reaction that they do. Sometimes they come from the reactant, in other words, the substrate that they interact with. Sometimes they have common names, okay? But sometimes if you are looking at names of biomolecules, one good indicator that you're working with an enzyme is going to be the ending. Many enzyme names end in ASE. So that's how you know, I was like, oh, that name ends in ACE. There is a possibility that it is an enzyme. So it is actually a good indicator. You can see here, like sucrase is an enzyme. Oxidase is an enzyme. In the case of the ones that have common names, then it's hard to know based on the name that it is an enzyme. But that information you basically memorize, okay? Now, it is important to note that enzymes can also be classified based on the type of reactions that they actually do, okay? So for the final exam, you guys need to know what will be the class of enzyme depending on the type of reaction. And you can see here the different classifications. Oxidoreductase is an enzyme that performs oxidation reduction reactions. Transferase is an enzyme that transfers atoms. Hydrolase is an enzyme that performs hydrolysis. A lyase is going to be an enzyme that add or remove atoms, but the best indicator is that when you look at the product, we're going to see a double bond forming or a new ring. Then we have isomerases, and isomerases is going to be the reactant and the product is going to have the same chemical formula. It's just that the functional groups are going to be different. So let me just write it here. So substrate and product have the same chemical formula, but different attachment of atoms. Typically, when it comes to that, if you compared the two molecules, you will say like, oh, okay, they have the same chemical formula, but it doesn't look like they have the same type of functional group. So that is an indication that an isomerase is the one that created the product based on the substrate. And lastly, we have ligases. Ligases are going to glue together two molecules, but the important thing is that you're going to see ATP as the source of energy for gluing the two molecules together. ATP is an important molecule that exists in cells that we derive most of the chemical energy that we see inside cells is via the use of ATP as a source of chemical energy. Now, when it comes to enzymes, let's note that, as I mentioned, the substrate, in other words, the reactant needs to bind a area that is a small region called the active site, and that's where the reactions are going to be catalyzed. That being said, since a reaction is happening, we can actually write in a reaction form an enzyme catalyzed reaction. In an enzyme catalyzed reaction, it has specifically to be written in this manner. You see E plus S, you see two arrows, you see ES, one arrow, E plus P. Let me actually state what all of those letters mean. So E is actually the enzyme. S is going to be the substrate. So of course, in an enzyme catalyzed reaction, we need enzyme and we need a substrate. Now, when those two come together, they're going to make what is called the enzyme substrate complex. Now, you may notice that when it comes to this, the formation of this enzyme substrate complex is a reversible process. The reason why it is a reversible process, which out of the two arrows, the one that tells you is reversible is the bottom one, is because like any other reaction, when you have things interacting in order to form product, they need to collide. They need to collide with enough energy to overcome the activation energy. And also they need to do it in the correct orientation. So when it comes to enzymes, those three parameters need to be met to correctly form that enzyme substrate complex. Once that enzyme substrate complex has been formed, then it can be committed into forming the product. As you can see at the end of this reaction, we still have E available. And we also have product form. It is important to note that enzymes are made at the end of the reaction. So they are actually utilized to take the substrate to product, but they are regenerated at the end of an enzyme catalyzed reaction. And this enzyme is able to continue the cycle for specifically the activity that that enzyme has. To give you guys an example of an enzyme catalyzed reaction, we have here a diagram that is going to illustrate to us a specific enzyme catalyzed reaction. So as you can see here, to illustrate the parts, we have our substrate, which is sucrose. We have our enzyme, which is sucrase. So when sucrose and sucrase come together, the substrate is going to bind the enzyme. Then the enzyme substrate complex is going to be formed. This enzyme specifically utilizes water in order to form the product. So water comes in to the enzyme, and then when it comes to formation of the product, we can form glucose and fructose as our products in this enzyme catalyzed reaction. So let me just write it in terms of how we illustrate enzyme catalyzed reactions. So for the example that I have here, the way that we will write it, it will be sucrase, sucrose is our substrate. The enzyme substrate complex will be sucrase, sucrose complex. Then in the products, we have sucrase by itself, and then we have glucose and fructose formed. Now, if we have to classify sucrase based on the reactions that we know in this course, because we are cutting something with water, understand that the classification of sucrase is going to be a hydrolase. Because we took a molecule, utilizing water, we cut it into two. And if we remind ourselves of the different types of organic reactions that we learned a few weeks ago, hydrolysis is how we cut molecules utilizing water. So this symbolizes that since sucrase was able to cut sucrose into two monosaccharides with the use of water, that's why sucrase is a hydrolase. Any questions on how we write enzyme catalyzed reactions? Then let's move on to the modes of actions for enzymes. Now, let's understand that enzymes need to meet the substrate, and there are two models that explain how that interaction actually happens. The first model that we have is called the lock and key model. And in the lock and key model, as you can see here, overall, what is occurring is that your enzyme is going to be acting as a lock, meaning it is a structure and there is an opening for it, okay? And then the key, quote unquote, is going to be the substrate, which means that the shape of that substrate is going to specifically fit in that pocket that has been designated for it inside of the lock. So you can see that the enzyme substrate complex for a lock and key model, the substrate has a specific shape for the active site. And overall, once they meet each other, then the product is going to be made. So this is technically the product. And as we learned, the enzyme is going to be regenerated. Now, some enzymes utilize what is called the induced fit model. In the induced fit model, understand that we have a substrate, but then the enzyme have what is called an active site that is relaxed, okay? So some parts of that active site will fit the shape of the substrate, but it is not exactly the shape of the substrate. In the induced fit model, understand that overall, that enzyme is going to have an active site that is going to be modified because it's going to adjust its shape and in a way hug that substrate in order to convert it to product. So out of these two models of action for enzymes, understand that the induced fit model is more dynamic because of the ability of that active site to be modified by the enzyme just basically adopting a shape that will accommodate the substrate. So again, enzymes can work in any of these two models. Now, the last concept that we're going to talk about when it comes to enzyme activity is specifically the factors that affect enzyme activity. There are three factors that affect the activity of an enzyme. Those factors are temperature, pH, or the use of inhibitors. And we are going to talk about each of them and how they affect enzyme activity. The first one that we're going to talk about is temperature. Enzymes need to work under specific temperatures in order for them to be the most active. So a designated temperature in which your enzyme is going to be the most active, it is known as the optimum temperature. The enzymes in our body need to work at 37 degrees Celsius. Okay, and you can see in the diagram that we have on the right side that the maximum enzyme activity, it is observed by looking at the top of the hill of that curve. Now, it is important to note that independently, if the temperature is increased, as you can see towards the right of the graph, or if the temperature is decreased, the activity of the enzyme is going to decrease in either direction. That's why we see that the curve is going down. Now, when it comes to temperature changes in the enzymes, let's understand that the reason why we see a decrease in enzyme activity is because the changes in kinetic energies that are happening. On the left side of our curve, as we see that we are reducing that temperature, the kinetic energy is decreasing, so that enzyme is not going to be moving as fast. The problem with that is that if the enzyme is not able to, in terms of its shape, be dynamic, that may affect its enzyme activity. On the right side of that curve, which is when we are increasing the temperature, understand that that can also affect the kinetic energy. But more importantly, what gets mostly affected when we increase the temperature is going to be the folding of the enzyme, which we already saw that under denaturation conditions, the higher the temperature, then if your enzyme, which is a protein, starts getting unfolded, it actually affects its activity. So understand that enzymes have an optimum temperature, and if you cool it, it affects its activity. If you heat it, it affects its activity. The next parameter is pH. pH also affects enzyme activity. Now, similar to what we saw with temperature, enzymes actually have an optimum pH. And what is an optimum pH? Well, the optimum pH is going to be the pH at which an enzyme is the most active. You can see here that this is only illustrating the optimum pH for enzymes that have a pH around 7.3 to 7.4. Now that is close to the pH of our blood. So understand that if the enzymes reside on our blood, that's going to be the maximum, their optimum pH. Okay, so let me just illustrate it here. So if they are in the bloodstream, maybe, possibly that is going to be its optimum pH. Now, this is the reality of optimum pH. Optimum pH for enzymes is not always 7.3 to 7.4. It depends on the location of the enzyme. Let me just write that note. Optimum pH depends on location. So to illustrate this, I have the following table. This is actually illustrating different enzymes, what is their location in the body. You also have the substrate, but it tells you the optimum pH. So I am not, asking you guys to memorize all this information. The main thing that I want you guys to know out of here is that optimum pH depends on location. We don't have a specific number that we're looking for. It depends on the location of that enzyme. So for example, here we have pepsin, which is an enzyme that is found in the stomach. When we were talking about acids and bases, understand that, for example, in our stomach lining, the pH is around 1 to 2 in pH value. That is acidic. But since pepsin is in the stomach, then it makes sense that its optimum pH is in the acidic range because that's where it is located, okay? Now, let's look at, for example, lipase, okay? Lipase, which lives in the pancreas, is involved specifically in the hydrolysis of ester bonds, specifically triglycerides. Notice that its optimum pH is 8.0. So, take-home message from this is optimum pH depends on location. We cannot say that all enzymes work under the same pH conditions. The last thing that we're going to talk about in terms of things that affect enzyme activity is going to be the use of inhibitors. The use of inhibitors has been created in order for us in the laboratory to actually understand how enzyme activity works, how the enzyme behaves in a particular environment, and is very useful if you're trying to find out, well, if the enzyme is not there, how does the environment all of a sudden actually modulates? So in the laboratory, and the use of inhibitors is really prevalent. In addition, there's actually some treatments out there that have been created in pharmaceutical companies that are inhibitors just because of certain enzymes that we may not want them to be too active on certain instances. Now, when it comes to inhibitors, understand that we, even though there's many types of inhibitors, we are only going to focus here in Chemistry 112 in discussing competitive inhibitors, non-competitive inhibitors, and lastly, irreversible inhibitors. In the following table, which I'm just going to write a note, have it for final exam. We have the comparison for competitive, non-competitive, and irreversible inhibitors in terms of the shape of the inhibitor, the binding or how it binds to the enzyme, and if it's reversible or not. So even though we have those descriptions, I want to utilize the figures that we have in the bottom in order to discuss the different types of inhibitors. So let's assume that this is the normal binding for an enzyme and its substrate. Yeah? We can see that there's an active site, and that active site has a similar shape to the substrate. We can see that the enzyme is there and is present. What if now to that enzyme solution, we introduce specifically a competitive inhibitor? A competitive inhibitor is going to have a similar shape, as you can see here, to the substrate. Since it has a similar shape to the substrate, that means that it can bind the active site. So in essence, the competition part of it is because now the natural substrate of the enzyme competes with this molecule that was introduced into the solution for the active site. Now, can competitive inhibitors be reversed? The answer is yes. How do we reverse them? Well, by adding more substrate to the solution, we are able to reverse the effects of these competitive inhibitor. Okay? Let's look at the next situation. What if instead of a competitive inhibitor, we have a non-competitive inhibitor? Okay? Now, non-competitive inhibitors, give me a moment. A non-competitive inhibitor does not have the similar shape. okay, to the substrate. Let's establish that. Interestingly, it binds away from the active site. When it does that, it actually changes the shape of the enzyme. At times, as you can see in this illustration, it may even change the shape of the active site. So the active site is going to be there, but the thing is that because the shape has been changed, now the natural substrate may not be able to bind. Because remember, as I mentioned a few moments ago, in order for an enzyme catalyzed reaction to work, that active site and that substrate need to collide, they need to fit each other. So a competitive inhibitor, again, modifies the shape of the enzyme. And now, even though you have an active site, it doesn't have the correct shape for the substrate to come in. Now, can non-competitive inhibitors be reversed? Well, you can reverse them specifically by chemical changing the enzyme such that the inhibitor can be removed. That's the way that we can actually reverse them. Lastly, we have irreversible inhibitors. And irreversible inhibitors do not have a similar shape to the substrate. Now, the reason why they are irreversible, so the changes that they make to the enzyme is going to be permanent and not reversible, is because that irreversible inhibitor is going to form a covalent bond with the enzyme. And that's how it achieves its type of inhibition. The classic example to explain irreversible inhibitor is going to be penicillin, that upon its discovery it actually changed the world of medicine because people were dying of bacterial infections. Then when penicillin was isolated and utilized, people were actually being recovered. But understand that by now, there has been people that have developed allergies to penicillin. So this is an area of research that continuous studies have to happen, because even some of the bacteria that were treated with penicillin, it actually developed resistance to penicillin. So understand that this is ongoing research that has to happen there. So with this concludes all of the topics that we have for the final exam. So thank you guys so much for coming. This is all that we have for today.